Good morning. Good morning. Nobel Prize laureates, excellencies, ladies and gentlemen, welcome to the 2024 Nobel Prize Symposium at the Embassy of Sweden in Washington. My name is Maria Brogren. Thank you. My name is Maria Brogren and I'm the Counselor for Science and Innovation at the Embassy of Sweden. Today we celebrate the scientific discoveries of this year's Nobel Prize laureates. It's a tradition here at the Embassy to invite the American or US-based laureates for a scientific symposium. And we are spoiled in that sense that there are usually at least a couple of US-based Nobel Prize laureates. This year, there are no less than 8 American or US-based Nobel Prize laureates, and 4 of them are here in person today. And my honor as the moderator of this symposium is to interview each of them about their discoveries in the fields of chemistry, medicine or physiology, and economic sciences. And I'm not an expert in any of these scientific fields, but I'm eager to learn as much as possible from our laureates. And hopefully all of you in the audience will get a chance to learn as well. At least that's my goal. And after my conversations with the laureates, I will open up for questions from the audience. And I will take one short question after my individual conversation with each of the laureates. And then in the end of the symposium, we will have questions for everyone. So you will all get a chance to pose the questions that I might not have asked. But first, before we dive into the details of the prize-winning science, I have the honor to introduce 3 distinguished speakers to set the scene and to provide us all with a background to why we are here today. So first, it's my honor to introduce His Excellency Urban Ahlin, Ambassador of Sweden to the United States.
Thank you very much, Maria, and warmly welcome to all of you, Nobel laureates, honored guests, and all of you following us virtually all around the world. My colleagues and I here at the Embassy greatly appreciate our long-standing partnership with the National Academy of Sciences and Dr. Marcia McNutt. It's a pleasure hosting today's Nobel Symposium together with you. Sweden is a knowledge-driven nation and places a strong emphasis on science and technology. Just a few weeks ago, the Swedish government proposed our largest investments in R&D in history, focusing on excellence in research and on innovation in emerging technologies. At the end of this year, Sweden will present a new research and innovation bill which will form the basis for the government's policy on research, innovation and higher education for the coming 4 years. R&D, innovation, engineering are a long-standing Swedish strength, with technology-based companies like Ericsson, AstraZeneca, Alfa Laval, Spotify, you can name lots of them. Sweden consistently tops international innovation indexes. And as I said yesterday when we had our Nobel Gala dinner, sometimes Switzerland is on the top and sometimes Sweden is on the top and so on. But as I said, it doesn't matter because people mix up Sweden and Switzerland anyhow. So we are always number 1. Sometimes we are called Switzerland and sometimes we call Sweden. But we are second to Silicon Valley. Stockholm is number 2nd. When it comes to successful startups per capita in the world, in so-called unicorn factory, actually only beaten by Silicon Valley. The economic ties between the United States and Sweden are strong, and according to the latest figures, Sweden is the 10th largest investor in the United States economy. It's not bad for a country of 10.5 million people. Trade flows between our countries are substantial, and the US is Sweden's largest trading partner outside of Europe. Alfred Nobel himself, he felt strongly about cooperation between nations. For Sweden, being a relatively small country, international cooperation is crucial. Transatlantic cooperation continues to be essential as we work together to address global security challenges, promote innovation and sustainability, and foster economic and technological advancement. My colleagues and I, we are continuing to trying to strengthening the collaboration between Sweden and the United States, bilaterally, through multilateral organizations like the European Union, and nowadays as an ally in NATO. To sustain technological advancements, we need technologically skilled workforce. A workforce that can advance knowledge, innovation, and problem solving. Governments worldwide can support the education of next generation of scientists and engineers. But young students also need role models. Dear Nobel laureates, you can play an important role in fostering a passion for science in youth, encouraging curiosity and resilience, empowering a next generation of scientists to tackle unknowns and push boundaries. I know we have representatives here from the National Academy's New Voices with us in this room today, and I hope you all take a chance later to ask the Nobel laureates your questions. Thanks again for joining us. I'm looking forward to learn more from our Nobel laureates about their extraordinary scientific achievements and their contributions to building a better world. Or, in Alfred Nobel's own words, scientific discoveries that provide the greatest benefit to humankind. Thank you.
The Embassy of Sweden is proud to co-host today's event with the National Academy of Sciences. And now I'd like to introduce Dr. Marcia McNutt, President of the National Academy of Sciences. Marcia McNutt earned her bachelor's degree at Colorado College and studied geophysics at Scripps Institution of Oceanography, where she also pursued her PhD in Earth Sciences. Building on that, Dr. McNutt is recognized for her contributions to marine exploration, where she has developed technologies to investigate processes occurring in the oceans and the seafloor. Dr. McNutt has held several leading positions at the intersection of science and public service. She has been the editor-in-chief of the journal Science and served as the director of the US Geological Survey and as the executive director of the Monterey Bay Aquarium Research Institute. Now Dr. McNutt is the 22nd president of the National Academy of Sciences and a strong proponent of long-term investments in research. Welcome, Marcia. The mic is yours.
Well, thank you, Maria, for that very kind introduction. And congratulations. There we go. And congratulations to all of the Nobel laureates. We are so proud of you. In my remarks today, you are going to hear me repeating many of the points that the ambassador just made. And I want to assure you that we did not coordinate our talking points ahead of time. And so you should take this as an indication that these really are important things to pay attention to. And I thought I'd start with just a little history about the Nobel Prizes. You've just heard that we have 8 American laureates this year. And that is a trend that we've been seeing for many years now. Since World War II, after Vannevar Bush wrote Science, the Endless Frontier, the US has dominated Nobel Prizes. We have dominated Nobel Prizes in a way that no other nation has dominated anything that keeps score. The US has 15% of all Olympic medals given in modern times. And at 15%, we are number 1 in the world. We're world leading. But that's nothing like the more than 50% of all of the science Nobel Prizes we've earned. So we should, as a nation, take immense pride in our science and technology enterprise here because it is not just world leading, it's been world dominating. But it wasn't always that case. Before World War II, if you took a list of who had the most Nobel Prizes, it was Germany. And what did Germany do to lose its first place? It did it to itself. It wasn't exactly competition elsewhere, but think of all the German scientists that left Germany in the ramp up to World War II, most of them Jewish researchers, top-notch intellectuals, but they recognized that their lives were going to be on the line if they stayed in Germany. And then after the war, there was a whole new wave of science immigrants from Germany who came to the US to continue their careers and to help the US with its rocket program, leading to its space program, the first moon landing, etc. But as we all know, Nobel Prizes are a lagging indicator of the strength of your research and technology environment. By the time someone wins their Nobel Prize, we've got some examples this year where the work was done just a few years ago, but very often the work was done a decade ago. And it takes that amount of time to truly verify that this is a new discovery that is going to benefit mankind. So that's understandable. But I'm worried that at the time we start seeing fewer Nobel Prizes coming to the US, we will have already lost our edge, maybe as much as a decade before. So I think it is really worth our while to think clearly about what are the attributes that make a nation a strong contender for Nobel Prizes. Well, I've got a little list here, and the first on it is immigration. About 28% of all Nobel Prizes won in America were first-generation immigrants. That's pretty amazing. I don't think you can look at any other high-level, except maybe the leaders of the Fortune 500 companies in America. Immigration has been critical to our success, and it has to be something that we continue to support and think about how we are going to continue to attract the best and brightest to our shores. Second thing, a good point that the ambassador made, is workforce. We have to, in this country, start worrying about the fact that when it comes to science education or math education in the US and we compare it to the top 20 nations in the world, economically and scientifically, we're middle of the road in terms of math and science scores by young people. And not all of those, just because we improve our education system doesn't mean that they are all going to become Nobel laureates, but at least we have to have the workforce that promotes the best and brightest and also helps turn all these discoveries into benefits for humankind. So I think the US has to put a lot more investment into our education system, starting with the very youngest children in elementary school, not teaching them facts, because science isn't about facts. Science is a process. It's a process that is constantly revising what we know and improving on it. And we have to excite children with hands-on discovery. Next thing I want to mention, which the ambassador already mentioned too, is international partnerships. I'm sorry to say that the US has kind of a reputation as a fickle partner because of our political system, which every time administrations change over, there are new priorities and new commitments. And when you look at some of the major facilities that are required today in order to make those great new discoveries, it used to be when I was a student starting out in high school and college, that an individual university would have a high-energy physics facility. Stanford had their linear accelerator, and other places had high-energy physics. Now, an individual institution couldn't create a leading capability to do new scientific research. And in fact, it's gotten beyond generally even the ability of a nation to do that. We have to work on these internationally, and the US has to become a better international partner. Next, of course, is investment. We have to continue to invest. I am thrilled to hear how Sweden is upping their ante and making sure that Sweden remains a top international powerhouse in science. I'm very concerned about the future for the US. I'm concerned because tax cuts typically lead to less investment by the country. And of course, we do have a problem with the deficit, but it is science and technology that is our antidote to the deficit. It is through investing in new technologies, new ways to create tools and new advances that we actually build our tax base to get out of the problem. And then finally, let me just end by saying perhaps the most important thing we can do, particularly here in the US, is address trust in science. We have to be a trustworthy enterprise, and we have to communicate better with the public about why they should trust science as well. Because without the public trust behind us, we're not going to make the investments. We're not going to attract the best and brightest from around the world. We aren't going to improve our science and math education in school. None of these things will happen without the public behind us. So while we're here to celebrate these amazing Americans who have won their Nobel Prizes, we also have to make sure that we can continue to do so. And I hope you will all join me in, first of all, congratulating them, but also recommitting ourselves to science excellence here in America. So thank you.
Thank you so much, Dr. McNutt, for your thought-provoking keynote. Really a lot of things to think about there. Now I have the honor to introduce Dr. Lars Heikensten, who will give an introduction to the great donor Alfred Nobel himself and the Nobel Prize. Dr. Heikensten received his doctoral degree in economics from Stockholm School of Economics. He is a former governor of the Swedish Central Bank, and he has held many international positions, such as member of the EU Monetary Committee and the General Council of the European Central Bank, and Swedish governor at the International Monetary Fund. He's also held numerous board positions in companies, universities, think tanks, and government agencies. But most importantly in this context, he served as the executive director of the Nobel Foundation for 11 years, with the ultimate responsibility for fulfilling the intentions in Alfred Nobel's will. Welcome, Dr. Heikensten. The floor is yours.
Thank you so much, Maria. Sitting beside Simon Johnson here, I realized that I should not have 7 minutes. I should have an hour to prepare him for the week in Stockholm. But if I should say one thing, be prepared for champagne. As was said here, I am not working with the Nobel Foundation now, so I'm speaking as a private citizen, so to say. But I realized when I put down a few notes here that I was writing we all the time. So I still consider myself part of it, but I'm not in reality, and you should know that if something is said here that should not be said in formal context. I'm a little bit unsure that there were pictures that were going to be shown. Should I indicate that and someone showing them, or? Yes, okay. So first I'd say a few words about Alfred Nobel, the man behind the prize. He was born in 1833 in Stockholm, and just after a year or so his father left him. He was an entrepreneur, and he got bankrupt, and he left the country and went to Finland, and then a little bit later he went to St. Petersburg. And when he was back on track, which he became in St. Petersburg, the family came along. St. Petersburg in those days was a major European cosmopolitan city. And Alfred grew up there together with his three brothers, and he got a very wide-ranging and good education. Later on in his life, he could speak and write 5 different languages. At the time, he was quite interested in poetry. Later on, he wrote a drama, but mostly he got engaged in sciences. And at a relatively early stage, he got in contact with nitroglycerin. This was a well-known explosive, but it was very difficult to control, so it couldn't really be used for practical purposes. But he got interested in that, and he tried all kinds of methodologies to deal with this, and finally he found a sand-like substance that he could mix with nitroglycerin and create a substance or that could be dealt with practically, and that he named dynamite. And that is what he lived on, essentially. But when one thinks about it, one should not think, I think, primarily about a scientist, but one should rather think about an entrepreneur. He built an international company, sort of, covering all of Europe and other parts of the world as well. And basically what he did was that he went to someone who had money in another country and he said, I can put in the patent, you put in the money. And then he kept at least 51% of the company, so he kept control and accumulated a lot of capital and could move on to the next country. But he had a scientific interest, and all in all he had between 300 and 400 patents. And many of them had to do with explosives and arms and so on. But he had also patents for bicycles and artificial silk and all kinds of things. You could take the next picture. He died in 1896, so he was 63 years old, in Sanremo in Italy. He died alone. He was not really having a happy life, and one can see that from his letter. He was a melancholic character. And of course, people knew that he had money, so there was a rush to find his will. And relatives came from Sweden down as quickly as they could, but after 5 days or so, it was found out that the will was safely placed with the Swedish bank, and it was opened in Stockholm, while some of the relatives were in Sanremo waiting for the result. And it came as a very big surprise. What was in the will, and basically it said that everything I have should be sold and should be devoted to prizes in physics, in chemistry, in physiology, in medicine, in literature, and in peace. And there was a strong reaction within the country because the prize could be given to other than Swedes. And this was very special at the time. There were good prizes, interesting prizes, valuable prizes in France and Britain, for example, but they never managed to get the status because they were only for nationals at the time. So that's an important ingredient in this prize, that it's a universal prize. And particularly irritating it was, of course, that the Norwegians got the right to give out the Peace Prize. That I don't think we still really comprehend the reason for. Norway at the time was in union with Sweden, though, so they did not have their own foreign policy. And that might have been a reason why Nobel chose them, but they also had a more democratic parliament than we had at the time, and that can also have played a role. But they certainly realized the potential. Suddenly, their parliament would get a foreign policy role through this prize. And I think it's correct to say that even though they have tried over the years to distance the prize from actual policy, it's still an issue from time to time. Not so long ago they gave it to a Chinese dissident, and that led to direct action on the part of the Chinese vis-à-vis Norway as a country. So it does have a political role, although, as I said, they are trying to move away from it, I think. As I said, there would be 5 prizes, and he pointed out the institutions that should be responsible for them, and that's the next picture here. And that is the Swedish Academy for Literature, the Royal Academy of Sciences for Physics and Chemistry, and later on the Economics Prize. I'm going to say a few words about that also later on if I have time. And the Norwegian Parliament for Peace and Karolinska Institutet for Medicine and Physiology. And they have they reacted a little bit differently. So some of them were, as I said, the Norwegians were eager to take on this task. Others were quite skeptical. And you have to remember that these all had their own history going back several hundred years. And the Swedish Academy, for example, primary role is really helping develop and take care of the Swedish language. So choosing literature on the world scene did not come very natural. But after a few years, the Nobel structure, the people dealing with the will, managed to convince this institution to take on the task. And they have, after this, after they did that, they have since 1901 given out prizes. And this they have been doing in basically the same way. And there are many issues, and Marsha McNutt touched on some of them. There are many issues here that I could elaborate on for a long time on how this process is being done and so on. But I think it's one of the most important factors with the prize is the legacy of how this job has been done. And let me end by saying that today the prize is 11 million Swedish kronor. That's roughly a million US. And that is what it has been corrected for inflation since the beginning, more or less. But of course, if you correct it for the price of apartments on Manhattan, it has not kept its value. So there is an issue here, I think, with the price. But and let me end with that a little story that when I was new, I was called up, and I regret I don't remember if it was Science or Nature, but it was one of the two who called me, and I was not so familiar with them at the time as I became, and they wanted to interview me about the new prizes. And I got quite worried because I thought that here I would come in new and there would be an article in the most important, one of the two most important newspapers or magazines for the scientific world saying that the Nobel Prize was under threat, and how could I cope with this? I didn't want that to happen. But then when I read the article, I got quite happy because the title of the article was The New Nobels. And then I figured that if that is the worst that could happen, that something become a new Nobel, then the Nobel is probably doing fairly well. And the article ended with a citation from the talk I had with them, where they said that, or where I said that that I think that it will be doing okay with the legacy it has, with the level of money involved, with the universality and so on. I think all these things would mean that it would do okay, as long as we did not make major mistakes ourselves. And that is, of course, to some extent what I had to deal with during those 11 years, to try to avoid that mistakes were being done. Those who followed the history know that a little bit better. But that's really where we are today. It is still considered the world's most prestigious prize according to Encyclopedia Britannica, and that I think we should be happy about. Thank you.
Thank you, Dr. Heikensten, for sharing your insights into the world's most prestigious prize. And now I have the honor to introduce our first Nobel Prize laureate. I will start with the Chemistry Prize to follow the order in which the prizes are mentioned in Alfred Nobel's will. The 2024 Nobel Prize in Chemistry is awarded with 1/2 to David Baker for computational protein design, and the other half jointly to Demis Hassabis and John Jumper for protein structure prediction. And now please welcome Dr. John Jumper on the stage. So I will just first start with a short introduction of you, and then you can correct me if I misunderstood anything. But
not if it's better than the truth.
John Jumper was born in Little Rock, Arkansas. I've actually been there. It's a very nice city, but I don't think so many have been there. It's quite small.
No, only a few 100,000. Yeah, the whole state is not that big.
Yeah. But it's very nice. He holds a Bachelor of Science with majors in Physics and Mathematics from Vanderbilt University, a Master of Philosophy in Theoretical Condensed Matter Physics from the University of Cambridge, a Master of Science in Theoretical Chemistry from the University of Chicago, and a Doctor of Philosophy in Theoretical Chemistry from the University of Chicago as well. And in 2017, Dr. Jumper joined the company Google DeepMind, where he started collaborating with Demis Hassabis. And in 2020, they presented the AI model called AlphaFold 2. And it is for this AI tool and its ability to predict protein structures from their amino acid sequence that they are awarded the Nobel Prize in Chemistry. And I should also say that in 2023, you became the director of Google DeepMind. So now you're in the leadership team, aren't you? Yes.
There are quite a few directors.
Yeah. So this is your current position?
Yes.
Yes. So to start with, you get the prize for AlphaFold 2. Can you explain to us here in the audience and those who are listening online, what is AlphaFold 2? What does it do?
AlphaFold 2 is an AI system. It's trained from data, and it essentially predicts an experiment that is done by scientists around the world. And the experiment is to determine the structure of proteins. So proteins are these wonderful nanomachines that our cells are made out of. They are a couple of thousand atoms. They are encoded by our DNA that tells the ribosome how to make these proteins. And when they're made, they spontaneously, according to the laws of physics, fold up into an intricate 3D shape. And this 3D shape, or at least many of them do, and this 3D shape is what is functional. And observing the 3D shape of proteins is incredibly hard. Proteins themselves are smaller than the wavelength of visible light, so you can't directly observe them under a microscope, or at least their detailed structure. And so we've done intricate experiments. These experiments take months to years. There are a couple of techniques, and importantly, these experiments have actually been done many times in science. So there is the Protein Data Bank, started by really a visionary group of scientists to collect these data from around the world. And at the time we did the work, I think there were about 140,000 protein structures available, representing an enormous societal investment. And so the work we did with our team at DeepMind was really to use these data to train an AI system. And it's worth saying this is not an AI system that can do what humans can do, but surprisingly fluently, this is a system to do what humans can't do. That if we need the structure of a protein, we have to go along this possibly years-long experimental path. And so we were able to develop a series of innovations in machine learning, basically developing the right types of building blocks and tools for machine learning on proteins in order to be extremely accurate, to get competitive to, not quite as good as, but very close to experimental accuracy on our computational predictions. And being computational, we're able to do this in about 5 or 10 minutes instead of a year. And because of that, scientists around the world have been using this in their research, discovering new interactions, discovering new biology. And it's really just been wonderful to see that, you know, the discoveries that have been made with the tools we've built.
Yeah, thank you. How do you validate that the result of AlphaFold 2 is correct, that it is doing this job?
Well, that's a really good question. One part of validation is easy. Every week, about 100 new structures go into the protein data bank. So it's very easy to check as it goes forward, is AlphaFold accurate for these particular structures? Possibly more importantly, and for building trust in this, a group of scientists, John Malt, Christoph Fidelis, and others, have actually run a biannual competition called CASP. And it started essentially out of some claims in the early 90s that protein structure prediction was solved, that we can basically get the right answer to within about an angstrom, and John Maltz has given a wonderful talk on this. And so they said, well, if this is solved, you wouldn't mind if we did a little experiment. We have some friends who have unpublished structures, and would you mind predicting them? And in 1994, people found out that they had tricked themselves, that in fact, this problem was quite a bit harder. And they continued going every 2 years and still going, doing blind predictions of structures. And so essentially all the major structure prediction groups from around the world would enter their systems and their own human judgment into making these predictions. And so we really knew very well, and they knew almost immediately when these systems were actually really good because they had these blind tests. And that's been an important part, and then later, of course, scientists using it in their practice. And I think maybe the final point is that being an AI system, the predictions are not going to be uniformly good. And so we developed a series of what we would call confidence measures, but the algorithm predicts its own error. How far would this be from the true solution if the solution had appeared in PDB? And this is trained at the same time as it predicts structure. And that's proven to be very, very effective at telling scientists, this is an answer, but maybe this answer is probably not right, or take it only with 2 grains of salt, and this answer is pretty reliable. And that's been an important, I think, part of the responsible uptake within the scientific community.
Thank you. And so for the sort of practical use, if the tool says that this has potentially a large error, so do the scientists, do they then go to experiments, or is that some way of using it in combination with more practical experiments?
So it depends quite a bit on the exact nature of what the scientists are doing. It's worth saying that quite a bit of the science that is done now, inspired by an AlphaFold structure, possibly scientists would have gone and tried to solve that structure, but they might have also simply decided it was too time-consuming for their research, and so they would proceed without structure and just have to get a little bit more lucky in the types of experiments they design. So I think it goes both directions, but it is in some cases AlphaFold can be low confidence, but a scientist can say this agrees with orthogonal data that they have. I think overall, scientists are very, very good at dealing with uncertainty. We all deal with kind of imperfect proxies of what's going on underneath and try and integrate that information into our science. So it really depends case to case.
And in the introduction, I stated that you have a physics background. So how come that you sort of shifted your career a bit, and also have you benefited from your physics background in the work that you are doing now on proteins?
Oh, absolutely. I very much still consider myself a physicist, and I've shifted by what physicists would consider a random walk. That I've really bounced. The way I ultimately I wanted to be a laws of the universe physicist at the beginning of my career, but the kind of time scales were intimidating. I remember sitting in a meeting doing some undergraduate research, and I said, this experiment we're working on, when's it going to turn on? And the professor says, well, I may be retired by then. And the professor next to him says, well, I'm a bit older. I may not be around by then. And I thought to myself, I need something with a bit more immediacy. But I did go to grad school in physics and actually dropped out of grad school. So the MPhil is actually me dropping out of a physics PhD program because I just didn't quite love what I was doing. And then I ended up working in a very fortuitous accident at this company, D.E. Shaw Research, which was studying how proteins move and building custom computer chips for that. And I fell in love with biophysics and really this notion that I'll study atoms and equations and all this exciting stuff that I think is fun and I think I'm good at. But then at the end, it'll be about whether someone leaves the hospital or not. And it has this direct and immediate meaning that was really appealing to me. And so I fell in love with that and then ended up going back to grad school in chemistry and then no longer having access to these incredible computer systems that had been developed at that company. And so getting into AI and try and do with algorithms what I no longer had computer hardware to do.
Thank you. AlphaFold predicts how the structure of the proteins, but for what are the applications then? Why do you want to know the structures?
So the reason you really want to know the structures is ultimately that we want to understand how to perturb the function of biological system, to understand it, to also understand how natural, for example, mutations or biological processes are affected. And one of the easiest ways to think about this is very often, and what AlphaFold is used a tremendous amount for, is you have two proteins, and they'll come together. And you want to make the smallest change to disrupt how these proteins come together, to understand whether this coming together is an important biological process that you might need to understand a disease, or you might need to understand a particular, say, worm phenotype. And so, AlphaFold predicts this. So what if I just change this to break right there, and then they no longer come together? And then you can observe the biological consequences, and that can help tell you about the function of those proteins and the function in which they come together. So it's really about tying the world of atoms out to the world of phenotypes. And what the structure tells you is how to perturb that system a little bit, how to change it, or how it works, or to understand. I think one beautiful example, there's a protein that's responsible for degrading other proteins. And they wanted to understand how that protein recognized the other proteins. And they would put these proteins together in AlphaFold, and they would see that two strands, a certain type of structure, would sandwich the other protein just like this. And there'd be a very small region of that protein which was in between. And then they could show removing that region in between meant that this protein was no longer degraded, that this was the region in which the levels of this protein are controlled. And that helped them understand a lot more data and how this actually contributes to the cell cycle and replication. And so we basically are trying to find a way to move from our understanding of molecules out to our understanding of organisms. And this provides a leg up and gives you the right idea to do the right experiments to understand this biology.
Thank you. I think that we need to move to a question from the audience. So yes, we have one over here. Quick question. Sorry, you need a microphone.
Kiana Ahlin, I'm a member of New Voices of National Academy and a professor at UCSD. I think what AlphaFold did for synthetic biology is amazing. Along with engineering proteins and gene editing, we can potentially surpass evolution. So what's next for you? Because there is one thing is missing, and predicting the enzyme function within the cell environment, which still requires some testing as cellular environment will change. So I'd like to know what your thoughts is about that. Thank you.
So honestly, I think there's an enormous number of things that should be next. I really do hope that in time we will see AI as kind of one of the first not AI, but AlphaFold is one of the first AI systems that deeply mattered from biology, but just one of many, many that did. But specifically to enzyme function, I think we are making progress. The latest version of AlphaFold, AlphaFold 3, predicts protein small molecule binding, and of course, this type of protein small molecule interaction is key to understanding mechanistically enzyme function. It remains to be seen exactly how far you can push this. But I think the other thing that we really ought to think a lot about, and one of the things I think about, is the most perfect AlphaFold, the completely accurate AlphaFold, will only tell us some about biology. There's so much more about the cell, about interactions, about the mesostructure, about regulation, that aren't directly even askable within the AlphaFold framework. And we have to think about how are we going to get out to cell interactions, how are we going to, right now we depend on incredibly bright scientists to look at AlphaFold structures and think about the wider cellular processes. How do we start to bring that into the domain of AI and machine learning? What are those problems going to look like? What data are they going to be based on? So I think I'm very excited in seeing how this grows outward in scale, hopefully getting us closer and closer to the phenotypes and the wonderful work, for example, the work on microRNAs that is so justly rewarded this year.
Thank you, Dr. Jumper. And with AI, I mean, this can really accelerate. And now we turn to laureates in medicine and physiology. So please welcome up on the stage. So we have Dr. Gary Ruvkun and Dr. Victor Ambros. And I will read a short introduction to both of you. Dr. Victor, I should first say that the 2024 Nobel Prize in Physiology or Medicine is awarded jointly to the both of you, Victor Ambros and Gary Ruvkun, for the discovery of microRNA and its role in post-transcriptional gene regulation. And Victor Ambros was born in Hanover, New Hampshire. He graduated from MIT with a bachelor's degree in biology and then continued at MIT for his graduate studies and his PhD in biology. During this time, he worked with David Baltimore, who won the Nobel Prize in Medicine in 1975. Gary Ruvkun was born in Berkeley, California, and pursued his undergraduate studies at the University of California, Berkeley, where he earned his bachelor's degree in biophysics. He then moved to the East Coast to pursue his doctoral studies in biophysics at Harvard University. Both Ruvkun and Ambros did postdoctoral training at MIT, working with Robert Horowitz, who won the prize in physiology or medicine in 2002 for his work on genetic regulation, using the tiny worm C. elegans, which you have also used in your research. And after a decade at MIT, Dr. Ambros joined the faculty of Harvard University, and that's where he conducted the research that led to the discovery of microRNA. Together with Gary Ruvkun. And later on, Dr. Ambros joined the University of Massachusetts Shan Medical School, where he today is the Silverman Professor of Natural Sciences in the program in molecular medicine. And Dr. Ruvkun instead joined the faculty at Massachusetts General Hospital and Harvard Medical School, where he made his contributions to the discovery of microRNA, collaborating with Dr. Ambros, so we should talk a bit about that, how you collaborated. And today, Dr. Ruvkun is a professor of genetics at Harvard Medical School and continues his research in gene regulation, aging, and metabolism. And Dr. Ruvkun and Dr. Ambros, can you describe what microRNA is and why is it important? Who wants to start?
Yeah, we...
Take it, Victor.
You go ahead. I mean, well, microRNA are very short RNAs, about 22 nucleotides long, which is unusually short. It was seemingly unusually short at the time that we discovered them back in the early 90s. And it turns out that from the collaborative work that we did, we found that the microRNA, the first one we found, was complementary to an M messenger RNA of another gene that it regulates. So our basic discovery was that the small RNA regulates other genes by base pairing to the mRNAs of those other genes. So you can think of microRNAs as a way for that evolution contrives to produce, evolves a gene that can sort of talk to the other parts of the genome by base pairing. And it has a particularly useful, let's say, application in so many different contexts because it's post-transcriptional and can be utilized to dampen noise or turn switches and so forth.
And it's fast. So much of what modern biology has been studying is transcriptional responses, how mRNAs go up and down in response to this or that. And there's a lag that RNA polymerase has to go to the chromatin, and it has to make an mRNA, and that has to get translated. And a microRNA regulating translation, the mRNA has already been made. It's already on a ribosome. And so it's a rather instantaneous response. And it's not a coincidence that microRNAs are sort of exploded in organisms with nervous systems, where the time scales on which things happen is pretty fast compared to a plant, where it's a 24-hour cycle. So there's interesting biology that it intersects.
I noticed that you sometimes say small RNA, but the prize is for microRNA. Is there, what's the definition here? How small are the microRNA?
Right, there's a very important context, which is RNA interference. And as it happens, I also worked with another Nobel laureate, Craig Mello, who was a graduate student in my lab. And Craig Mello received the Nobel Prize along with Andy Fire for discovering, using C. elegans as their research system, so-called RNAi, or RNA interference. And that is a mechanism that is very closely related to microRNAs, because it uses the same protein partner, it uses the same kind of small RNA of about the same length. But RNAi, as discovered by Mello and Fire, is a mechanism by which the cell detects foreign RNAs that are coming, for example, from a viral replication. And that RNA is recognized by the cell as foreign, and it's chopped up into little 22 nucleotide pieces. It associates with the same kind of argonaute protein that microRNAs associate with, and then it silences targets by destroying them. And the microRNAs, though, at least in animals, are, again, the same protein, argonaute, the same small RNA, but it comes from the genome, from a part of the genome that's evolved to basically enter into this RNAi pathway. And also, go ahead. Yeah.
But there was a magical convergence around 2000.
Yeah.
So 1999 is when RNAi is discovered. By mistake, and a beautiful interpretation that they figured out that it was double-stranded RNA that did it. Yeah. And then David Balcom in plants discovered that in RNA interference, there's 22 nucleotide siRNAs. And for us in the micro field, which there was just two of us in the field at that moment. It's a small group, right? We're like 22. Oh my God! Yeah. And I brought people into my office because I found that that there's a whole Kabbalah tradition of numerology.
Oh no, the
Jewish
22.
22, and they're like, Jesus, he's gone nuts.
Yeah, right.
So I had like New Age music playing in the background.
Yeah.
But it was 22, and it was a magic number for us. And that's just, it's 22 something 22 long can find one spot in a billion. So it's just the right amount of information for base pairing to have specificity.
Yeah, and the amount of base pairing, at least in animals, is not the whole 22 nucleotides. It's mainly part of the beginning and then some towards the end. And folks have pointed out that this is, first of all, the microRNA does not destroy the target necessarily. And so that evolution now can shape the outcome and tune the amount of regulation that microRNA exerts when it binds. And also the interaction between a microRNA and its targets is evolutionarily very fluid, because it's easy, so to speak, to acquire complementarity to just a short part of a microRNA. And now selection can work on that, right? And it's also easy to lose that by just a simple nucleotide chain. So evolution is, we think, so you can think of it as always kind of interrogating the microRNAs and their target repertoires. And we see that those repertoires can be very fluid in closely related species. But at the same time, some of the interactions are deeply conserved. So my colleague's LET7, which you just got, is completely conserved in sequence, and there are certain targets that it regulates in all the animals. So we know that our common ancestor, LET7, was diligently doing something really important on these certain targets, Lin41, Lin28, and it's still doing it today in most of us, and so it's a wonderful sort of a dichotomy, I think, this evolutionary fluidity of the networking of the microRNAs, but at the same time, some interesting hardwired, almost permanent, sort of fundamental microRNA target interactions, and we don't really understand.
We were aided. The genome projects came to fruition right at the moment when we're working on this. and it made the activation energy to make inquiries. Well, is this conserved? You know, it was a 9600 baud modem from my desktop at home, right? Looking for LET7 in humans. You know, you get back the data. There it is. You know, it's faster now, but we had the Genome Project at our fingertips, and this was money well spent by governments around the world that millions of us have engaged.
Yeah, there was a very interesting international cooperation going on, because in Britain, John Sulston and Alan Coulson were putting together a physical map of the C. elegans genome, anticipating that they were going to sequence it in a few years. And those data that they were creating daily, or weekly anyway, would be posted in the Worm Breeders Gazette.
Well, and they would focus on the areas we asked them to.
Right, and then
you said, hey, we want to get to this gene, and they would work on our region.
Right, and they would send us data or clone pieces of DNA, and then we would, Candy's in the back, you know, she was mapping sequences near Lin4, and she would send back data to Alan and John, and they would send new clones, and they were doing this pro bono. They're not official collaborators. They were doing this with dozens of labs around the world at the same time. So this community was being built in a way around the genome, and the catalyst for that was John Sulston and Alan Coulson, and Sulston is also a Nobel laureate. You know,
but by the way,
for his work on C. elegans,
he came to visit Bob Horvitz's lab, and he watched us when we're postdocs, and he said, you know, a mind is a terrible thing to waste, you know, because we're doing this boring chromosome walking, and he goes, I can do this in parallel from 500 places, because we're doing it from one place. He goes, yeah, I'm going to save these assholes, and he, you know, and he did it. It wasn't just us; it was you know, hundreds of other people were doing that.
It was like crowdsourcing of.
Yes,
right. Yeah.
But he was so nice.
Yeah, and he, this man was.
Well, he's just. Well, we're charming.
He was at the MRC. No. Yeah, I mean, he was a leader. He was defining this. He came and gave a talk at MIT, and his first line was, I'm interested in building a community around the genome. That was like his first line. And I'm in the audience, and I say, what? You know, and this was his vision was, and it's true. I mean, the best scientific communities, and we think we're in one of them, the C. elegans community, or the RNA community, is built by people who have a vision and set a tone. And Andy Fire is another person who set the tone. So we would like to shout out to the fact that we're here because of a community.
And we're role models now.
I know. Yeah. That's the cognitive disconnect.
Thank you so much. I'd like to open up for one question from the audience now. We have one here.
Hi, my name is Monica Deuce. I'm a professor at the University of Michigan, and I'm a member of the NASA New Voices. I actually did my PhD in small RNAs, and the paper from 1998 inspired me to go to graduate school.
Yeah.
So one of the things I find super amazing about the discoveries of mRNAs is that I feel like it's not just its potential for medicine and humanity, but it's like discovering a new force of nature on which genes are dialed up and down. And you talked about the element of time, and I think as a neuroscientist now I think about the element of space, because they can turn things on and on a meter down the axon of a neuron. And the thing that's really amazing conceptually is that it was really a conceptual discovery, I think, because you said you did it through your like old computer at home, and so the technology was important, but it was really about looking at biology in a different way. And so I'm wondering, what other things do you think we're stuck on, like DNA and proteins, like we were stuck on, and now the word of RNA opened everything. So what other things do you two feel that we're being dogmatic about science that we could look beyond and open new forces? Thank you.
Ignore the cytoplasm. The membrane proteins are where it's at.
Okay, that's the latest.
That's me. It's my advice to you youngsters. So, and this is like a new passion, but you know, like my favorite protein of all the, you know, there's 20,000, ATP synthase, right, which is the membrane passing, the thing that pumps protons by doing this. It's the best
Yeah,
protein. It's the best protein ever. You know?
Okay, I have two parts to the two-part answer to your question. First of all, is listen to guys like this, right? Because he has new ideas, new passions. They come and go, but each time when he explains it, you realize, oh, yeah, I hadn't thought of that. That's pretty amazing, right? And your record of you know is right out there of just ideas. You know, and so find people who are willing to brainstorm, you know, and the other thing is, I think the key is we don't know what we don't know, right? And so, I think that goes against human nature, right? Because I feel like when we interact with our everyday world, it's very important to stay sane, and to stay sane, you have to assume we kind of understand everything that's going to happen. If I do this, I do that, I walk this way or that way, you know, and so we're conditioned to think we understand most of what we need to understand, right, to function. But in science, so I feel that, at least it happens in my mind, you constantly have to deliberately work against the instincts to assume that we know everything we need to know. And that the thing is, we've discovered so much, you know, as a history of the species, you kind of think, geez, this is incredible. We must be almost there. There can't be that many more mysteries, right? I mean, can living things really have more fundamental things that we never would have thought of? And I think I feel we have to learn to have faith in that idea that there are almost infinite number of mysteries. You know, I mean, when you think about it, you got to stop me.
Yeah. I think your prize and your research is really proof of that. That there are new things to discover that you didn't think of before.
But it's happening in every lab. You know, in my college, you walk down the hall. It's happening in each of those labs now. You know, it's often we're not sure what they found, but they're finding stuff that's really surprising all the time.
Thank you. We will come back to you. But now I will turn to the economic prize. The prize, it's called the Sveriges Riksbank Prize in Economic Sciences in memory of Alfred Nobel. And it is awarded jointly to Daron Acemoglu, Simon Johnson, and James Robinson for studies of how institutions are formed and affect prosperity. And Dr. Simon Johnson, that is here with us today, he was born in Sheffield, United Kingdom, and holds a master's degree in economics from University of Manchester and a bachelor's in economics and politics from Corpus Christi College in Oxford. Johnson completed his doctoral studies at MIT, earning a PhD in economics. And after his PhD, he was a postdoc fellow at Harvard University. He then joined the faculty at Duke University. And in 1997, Johnson joined the MIT Sloan School of Management, where he has been a professor of entrepreneurship since 2004. He has also served as chief economist of the International Monetary Fund. And Dr. Johnson's research has focused on the impact of institutions on economic growth and development, and he is awarded for studies of how institutions are formed and affect prosperity. So, Dr. Johnson, can you tell us a bit more about your prize-winning research?
So, I spent about 10 years after my PhD working in Eastern Europe and the former Soviet Union. The ambassador spoke very eloquently at dinner last night about the hopes and aspirations that we all had for that region and the hard work that was put in by many people in those countries. And for me, those 10 years were extremely informative and also deeply frustrating, because many of the conventional, let's say, economic tools that I'd learned in graduate school were useful, but not really as helpful to me or to the people of those countries as I thought they would be. And I began to understand problems around corruption, the underground economy, and how political systems worked, and the way that was of first-order importance for macroeconomics. And then I met Daron Acemoglu and Jim Robinson, and one fateful conversation in 1999, Daron explained to me some of their thinking about broader global economic history, including what Europeans had done around the world when they essentially took over, colonized, and heavily influenced almost every society that exists today. And Daron explained some of the progress they've made on the theoretical side, but there are a number of big empirical questions, including whether we could solve or move the ball in any way with regard to causation. You know, was it a question that countries became richer, they got better institutions, or something about institutions being created early on that drove what happened later to GDP per capita? And in that conversation with Dron, I realized two things. First of all, that this was an incredibly hard question, one of the hardest questions in economics, and almost an almost impossible task to crack the empirical side of this. And the second thing I realized was this is what I wanted to work on flat out for the foreseeable future. And we were very lucky to make some progress ourselves and to open the door to other researchers. Economics is not science, let's be very clear about that, although I think the economics work that we do is very pro-science and very much explaining the importance of science and how we've built some societies that are very pro-scientific and why we should do a lot more to foster science. But the key thing for us is that a lot of other scholars walk through the door, and the doors that we opened, particularly linking the importance of history and understanding deep history and quantifying that and linking that to the modern experience. I mean, we don't think that data solves all the questions. We don't think by any means we've answered entirely the question of causation in all situations, but I think we have helped reorient part of the economics profession towards that perspective on economic research and historical inquiry.
Can I ask you a bit more about the data collection? I know I didn't prepare you for this, but I mean, what kind of data, and how do you did you have like a framework that these are institutions and these are the sort of the monetary terms, and how did you, so what kind of framework for the empirical part?
What we were really trying to understand, so many people had done work around this area before, so we were standing on their shoulders. But what we were trying to understand is why the Europeans did such fundamentally different things in different parts of the world. So within the British Empire, there's variation from this, for example, which is an incredibly innovative society with, for sure, problems to be addressed, but quite a remarkable achievement in terms of what we've been able to build in the United States, to countries that are very far from us in income per capita, including in South Asia and including countries in West Africa, for example. And what we realized and discovered was that a large part of the European motivation stemmed from what the Europeans believed they could achieve for themselves. It's a pretty self-centered, I have to say, narrow or even selfish view. And that was determined in part by the mortality that Europeans faced when they traveled to different parts of the world before the germ theory of disease, before any progress had been made with regard to understanding tropical disease in particular. And the basic fact of the matter is that if you sent 1,000 Europeans around the world in 1800, in this, let's say, the formative colonial phase, you sent 1,000 Europeans to West Africa, about 500 of them would die in the first year, mostly from malaria and yellow fever, which they had no immunity and there was no treatment or prevention. If you sent 1,000 Europeans to India or to the Caribbean, between 80 and 120 of them would die. If you sent the same 1,000 Europeans to the northern part of the United States or Australia, fewer of them would die than if they stayed behind in Europe. So the mortality they faced, the incentive, was dramatically different, and they therefore, in some places, established more inclusive to Europeans institutions. In other places, they established extremely unpleasant and nasty extractive institutions that favored the Europeans, but the Europeans were not going to go there in large numbers. They were going to extract gold, tax resources,
for
or
more
slaves.
short-term sort of thinking, or
that's one way to think about it. But remember, the slave trade was not a short-term business. Slave trade operated for hundreds of years, but it was short-term in the sense that the Europeans did not plan to move there. They wouldn't take their families. They weren't going to live there. It was get in, get out, make money quick, and they established rules of the game that favored that, sometimes directly and sometimes working with local people. And the very dramatic and in many parts of the world sad facts are that those rules of the game have lasted. There's legacies that exist today. It's not exact persistence, of course. Things do change over hundreds of years, but there's a legacy, an imprint on those societies that the Europeans placed there, and many places in the world are still today, 200, 400 years later, trying to escape those legacies.
So why is it so hard to escape those legacies?
Well, again, the extremely unfortunate and difficult truth is that some people do well when you establish extractive institutions, a relatively small elite. Now, that elite was Europeans during the colonial phase, but when there was a transition to independence, which has happened obviously almost everywhere in the previously colonized world, local people who took over found it advantageous to themselves to operate some version of what the Europeans had previously operated. Now, it's not exactly the same, but it's that persistence or that path dependence that has had a huge effect on these societies and that has really shaped the distribution of income and global inequality that we struggle with today.
So have you identified any countries or any models that can shift the institutions to become more inclusive? Are there some good examples?
Sure, there are certainly places that started with difficult institutions and a lot of poverty in the modern period. South Korea, my wife's family is from South Korea. I spent a lot of time thinking about it and studying it and discussing it with them. And they came up from extreme poverty and a very difficult institutional legacy in the 1950s. And through a great deal of struggle and also engagement with the world on their own terms in positive ways, and by the way, encountering certain crises that they had to navigate their way through, they have found their way to a better position in the world and a higher level of income per capita, although they themselves will tell you that the job is not done and there are plenty of problems that they face. So we do have certainly exceptions to the rule, and that's very encouraging, but we can also see many countries that have attempted to get onto a better path and even had a good run for 10 years, 20 years, sometimes 30 years, and then they have some reversion back to that difficult, essentially European colonial legacy.
And the reversion, is that because there some people sort of start to figure that this doesn't gain me anymore, so there is opposition, or why is it difficult to continue on the track?
Well, there's a number of explanations in different situations, but the key problem, again, the ambassador talked about that in his speech last night, is if you have an economy that produces a middle class, the middle class becomes prosperous, and the middle class is willing to share the wealth with the working class so that wages start to rise, then you tend to get a strengthening of the institutions. You actually tend to get the tilt towards science that's produced this fantastic research today, and you have what we regard as modern democracies, for example, the West European variety. But if it's the case that the money that's made is from, for example, natural resources, oil boom would be an example in modern Russia, again, the ambassador spoke about that yesterday, then that enriches a small number of people who are not inclined to either have a broader middle class or to allow voice or to allow wages to rise. And what you're basically doing is strengthening people who are benefiting from the extractive institutions. So, not surprisingly, they are not enthusiastic about building something that is more inclusive. And again, interestingly, what we see over time is those societies do not go so heavily into science, do not certainly reward the kind of participatory, citizen-led, decentralized science that we're celebrating here today.
Thank you. And just one more question for you from me, and then we will open up for questions from the audience. But you actually got your prize for something. The sort of main papers are from 20 years ago. So what path have you taken after that?
Yeah, the work that we did, that the prize committee regarded as seminal, was done 25 years ago. And we have not been, and we worked I would say, following the initial breakthrough, we worked very hard on that for 5 years, and then another 5 years of follow-up and responding to critiques and extending results and so on. But since then, we've spent a lot of time on technology and development of technology and understanding what has happened to the middle class. As Marcia said in her remarks earlier, if democracy fails to deliver for the middle class, you lose credibility, you lose faith in the institutions, and that actually undermines the support for science and the funding for science in a way that will further deplete your ability to innovate. So even though, for example, the United States has achieved remarkable things, I think this is by far the most innovative society in the history of humankind, it is all now, sadly and quite literally, in jeopardy, because over the past 40 years, technology has taken a path that has hollowed out the middle class, essentially through a lot of automation. Globalization has, to a large extent, reinforced that process, and the decline of trade unions has, I'm afraid, been most unfortunate in this regard. So people at the top end, people with a lot of education, including, I would think, most people in this room, have done very well as technology has developed. It's called skill-based technological change in the economics jargon. People in the middle have come under great pressure, and many of those middle education, middle-income jobs have disappeared. They've got pushed down to the lower end of the labor market, and the dramatic, really dramatic fact that's established, for example, by my colleague David Autor at MIT, one of the world's leading labor economists, and whose name I will be dropping in Stockholm as much as I can. David and John and I run a research center on this, but if you look at the real weekly earnings of men in the United States who did not go to college, it has not increased since the mid-1960s. So why they ask the question now, which is an entirely reasonable question. If you're putting this money into science, if you're having these advances, if these geniuses here are so far pushing human knowledge and we commercialize it, we take it to market to a remarkable degree in the United States, and we influence the world with this technology, why has that not benefited ordinary working people? Why has the middle class come under so much pressure? These are entirely reasonable and legitimate questions that demand an answer, and that answer has not yet been provided by our economists, by our scientists, by our policy apparatus. So my reaction to the recent election result and to all of the debates we've been having is very simple. There is a lot of work that we need to do.
Thank you.
Well, Nobel Prize, I think my colleagues need to understand, Nobel Prize winners have an elevated position in global society. Every word you say is being taken down and turned into theses and controversies around the world. So keep at it. It would be my advice.
Everyone will ask you questions about your science, but also about anything else.
Things you don't know anything about.
Yes. So do we have one question for Dr. Johnson? Yes, please.
Hello, my name is Alyssa Rudelis, and I'm a AAAS Science and Technology Policy Fellow and a physicist by training. My question is, do you see optimism in the institutions that we're currently building to address some of our biggest challenges in society, like climate change, of how those institutions can be built in better ways, more sustainable ways to increase prosperity internationally? Thank you.
So I had to leave the room for a few moments a little while ago to call Vegas and place some bets on John Jumper and how many Nobel Prizes he's going to win. Because he's a physicist who just won the chemistry prize, and he was explaining how he's going to transform biology. So we got those locked up, and so on. I think that we're in a serious answer to the question. We're at a very important moment and juncture. So the arrival of this new technology, AI, does provide us with incredible ability to move knowledge forward. And AlphaFold is, I think, by far our most dramatic example, but it is extremely dramatic. But the problem is, so this is. I'm being optimistic here. There's a lot that can be done with the advance in knowledge, and the key thing from a labor market perspective is, do we create new tasks for humans that require expertise? That's why you get paid more money than somebody who's just hired off the street. That's why I get paid more money, because those things are valuable to someone who has the ability to pay, to a market or to the government funding agency and so on. The problem is we are encountering this technology at a moment when we've already had 40 years of increased inequality, driven in large part by what technology, the way digital transformations took place. Now, I'm not pointing any fingers. I'm not saying it was anybody's fault or responsibility or there's any kind of conspiracy. That's just the way it played out. But it has played out in that way. So we've already much more. If we were having this discussion in 1980, I think we'd say, great, technology's arrived, strong middle class country, go for it. Good things will happen. And it's America, so, I don't know what's going to happen. We'll sort it all out.
It'll be good, though, yeah.
It'll be good, right? That was the optimism. In 2024, we've got to think about it a bit more, and I think we have to engage with scientists and engineers and everybody, including in this room, and think about exactly what we want to invent. There's a purposeful feature. What kind of jobs we're going to create for ourselves and for others, which problems we're going to solve, and how do we prevent both the intended and unintended consequences that come from hollowing out the middle class. Now, Kurt Vonnegut wrote a brilliant book about this. It's called Player Piano. Wrote it in 1949, I believe. He was thinking about cybernetics, one of the pre-runners of AI, and he predicted, or science fiction forecasted, a society in which there were a few highly skilled people who ran complicated engineering plants and all of production, and a lot of people who had make work. He didn't call it universal basic income, but it was. And that second group is extremely disaffected, really disillusioned, has lost the dignity of work, and has become extremely alienated, and they're one step away from crazy populism. Now, I do say to all my audiences, if you're only going to read one book after hearing me speak, you should read my book. It's called Power and Progress. and I've got 30 copies for free downstairs if you rush down there afterwards.
What if it's 2 books?
Yeah, hold on. My book is 500 pages long, and it's perfect for keeping your relatives quiet at Thanksgiving. Here, read this, Uncle so-so. Yes, but absolutely, absolutely, as Victor said, if you're going to read 2 books, read Kurt Vonnegut's novel. And I said this on a podcast recently, and I hope you've all been overwhelmed with podcast requests as I have. And the podcast host later showed up at my office with a signed print from Kurt Vonnegut. I didn't know it existed. And it's a line from Play a Piano, which is now hanging on the wall of my office, and it'll be in central place going forward. And at a key moment, the PhD engineer asked the computer, which is running the engineering works, what are people for? And that was Vonnegut's question in 1949. I think that's the question before us now. And I think, what are people for? What are science for? For whom are we inventing today? And we have a lot of knowledge about how invention goes well and how invention doesn't go well. And the Nobel Prize Foundation has had an amazing contribution to advancing knowledge and promoting brilliant ideas and some rather dubious choices that we need to discuss in Stockholm too. And I think we need to learn from that and think about how to get us all onto this more positive path, which is absolutely accessible. We call it pro-worker AI. You have to have a bumper sticker for what you want, in my experience. Like pro-worker AI is what we're pushing for. And it's absolutely attainable, but it's not, I think, our default path, and not the path we will fall onto automatically.
You have to be deliberate about it,
Yes, exactly. See, I got that from a Nobel Prize winner, which I'm now, it's been broadcast around the world. Victor says, you've got to be deliberate. So that's for you.
Sorry.
Yes.
No, sorry, that's perfect.
Yes. So I was going to ask you, but now we sort of we're sort of taken away here. But so if we back down to when you were younger, so what really spurred your interest in science from the beginning? I mean, now you're here, but what was the thing that got you started on this path? If it started you, John.
I think I was in love with this notion that we would find the truth at the bottom, that there was kind of quantum mechanics and relativity were my heroes. Even more, not exactly Einstein, but just this, Solvay conference, the early times in which, the modern world was discovered, the technologies, what we're all kind of built out of. And I was honestly a little bit biology ignorant at that point, and I thought it was a bunch of weird accidental complexity that I wasn't interested in. But for me, it was really about that moment where we really found what was going on in the world, and I love that. And I also love the kind of, notion of the kind of, we obtain this knowledge that we almost have no right to. It's so small, it's unobservable. It's the origin and fate of the universe. What right do we have? Like the TV show, we're on the third rock from the sun. We're on a planet in an unimportant solar system and an unimportant galaxy, and we're talking about what happens to the universe at the beginning and the end. And I just love that notion that we as people, as humans, could access that, could think about it, and it felt very rarefied, like the secret knowledge that you had to work very hard to even understand what they were saying. And then, none of these are really good reasons, but they're a lot of fun.
Yeah.
Gary.
Yeah, learning how does the sun work? The cycle of fusion in the sun, and they figured that out, 50 years ago, and hearing that when I was in college. And, I went to UC Berkeley. My tuition was $600 a year, right? So this was supported by the state, visionary for what it did to transform California into a major world economy. And, my professors in the physics department, you could just tell you were seeing magical minds that were presenting in physics 4D, the freshman physics course, and that's the show. When you refer to baseball, there's the minor leagues and the major leagues. The major leagues they call the show. Well, that was the show, and I'm 18 years old, and I'm like, that's what I want to be like that. And it's not just science. I had history professor, Leon Litwack, the history of African Americans in the US, unbelievable history course. You know, but it teaches you, this is what it is to be an intellectual, this is what it is to study society carefully and to do exposition about it for an hour and everybody's listening. You know, this is like magic. So that taught me where to go.
Victor.
Yeah, I mean, I can remember exactly this learning, reading about scientists and being so captivated by the fact that they figured something out that they kind of, like you said, they had no right to figure out. Because like I remember Ole Romer, right, figured out the speed of light back in the 16-something or other.
Was this the two mountains in the mirror?
No, he's observing the eclipses of the orbits of Jupiter's satellites and timing them. And of course, as it's going away, there's a certain kind of lag, and so it's coming back a different kind of lag. And you can see a painting of him at his telescope, and there's a pendulum clock. So he's timing with his pendulum clock. And he calculated the speed of light, and it was not bad. It was pretty close. You know, so that really, I think, wow. You know, you can do that just by observing, counting, writing something down.
And thinking.
You know, so yeah, that kind of thing. And I felt like I wanted to be part of that. I wanted to be part of that business. I want to be able to say I was a scientist and I was kind of like part of that, you know. And that's all I wanted to be, was, you know, just to be doing that kind of thing.
So it just turns out if you win a Nobel Prize, you're invited to donate one artifact to the Nobel Museum. Mm-hmm. And I'm going to donate my grandfather's passport that was issued in May 1942. My grandfather was a metallurgist. He had a PhD in metallurgy. He's the first person, I'm the second person in my family to complete college after my grandfather. And he was an expert in steel and ran a foundry in Sheffield, my hometown. He also passed away 2 weeks after I was born, but his life and his perspective was somewhat legendary. And my mother in particular emphasized the importance of education because of what she learned from her father. But in May 1942, he led a mission from the UK to the United States. And we have the visas issued, we have the entry and so on. And I believe that he went to Pittsburgh, possibly also Cleveland and Chicago. And my father, who's 89 years old, and I have thought about this quite a lot, and we are quite confident, both from the family history and also other documents and the time, that the British did not come to the United States to learn from the Americans how to make better tanks and tank tracks. They came to teach the Americans what they had learned, because the British had been building tanks and working very hard on their armaments industry for at least 3 years. The Americans had built almost no tanks before Pearl Harbor. Yeah. So this was a moment, and this wasn't the only moment, but this was a moment when the UK, in utter desperation and gratitude, turned to the United States, which was an amazingly powerful engineering country and industrial production country, but as Marcia said, was not a scientific powerhouse before 1939, as reflected, for example, in Nobel Prizes. The moment when technological leadership passed finally and irrevocably, or at least to this date, to the United States was in 1942, when the Americans did things like they took the cavity magnetron, which was actually delivered by the Tizard mission in end of August, September 1940, and they turned that into radar you could put into the nose of an aircraft to detect submarines in the Atlantic by early 1942. That's why the Americans and their allies prevailed in the Battle of the Atlantic, and shipping could go through to Europe. So to me, my grandfather's passport is immensely important personally. And that story, and also what happened to Sheffield, which went through a long period of industrial decline. The north of England also has struggled. But it's that, to me, what has, I think my entire career has pulled me towards the question of the power, but also the responsibility that comes with scientific leadership. What are you building, and for whom? And how is that prosperity shared across your country, in our country, the United States, but also around the world? Is this something that is genuinely more inclusive? Does it pull everyone up? Or do some people get pushed down while others are rising? And why? And how do we avoid that?
Sharing radar was huge.
Yeah.
Absolutely huge.
Yeah, the British.
Well, didn't you both work in the building that had previously been the Rad Lab at MIT?
Yeah, I did, yeah. Yeah, I worked in Building 20 for a while. Yeah. But the British brought it. and presented it to the states, right? And said,
So
this is what we can do.
It was done. The conversation took place at the Wardman Park Hotel, a couple of miles from here, and the British brought all of the things they thought, all the things they'd invented, and all the things they thought were going to change the war, and they showed them to the Americans.
Yeah.
And the Americans like, oh no, we already know that. No, that's not interesting. And then they showed them the cavity magnetron, and they did a few tests on it actually. But their initial reaction, which proved to be exact on target, was, oh my goodness, this is going to change the world, and it did, right?
Fantastic.
Including not just that piece of technology, but the innovation process that it triggered. And just to remind everyone, the moment, 1940, 1941, the topic they were working on was radar. But they wanted to keep that effort secret, including from Congress, I think, a little bit, because of the circumstances. So they put a code name on the laboratory, a code name that would clearly be something that was not weapons related. They called it the Radiation Lab. Right? That was the code name to disguise work on weapons in 1940.
Yeah.
Just to show you the acceleration. And as Vannevar Bush and the Endless Frontier report has already been mentioned, including by Master several times last night, that the key conceptual breakthrough at the policy level and at the engineering level, the science level, was to say, look, World War II, the US and its allies prevailed in World War II in part because they were able to apply previously existing scientific knowledge to the problem of war. But clearly, what you need for national security and competitiveness and prosperity, but first and foremost, national security, it's not a stock of knowledge. It's not a stock of aircraft. The largest air force in the world in 1923 was French. In 1929, it was all obsolete, right? What you needed to defend yourself in 1940 was aircraft engines that allowed planes to travel at 500 miles an hour. And those were not the engines of 1936 or 1921. They were engines that you were inventing and you could improve at that moment. So it's the ability to innovate and solve problems, and it's the money and national effort we put into the scientific enterprise after World War II that tipped the balance and the Nobel Prizes in our favor. And the challenge of Sputnik, which was launched in October 1957, was met in bipartisan fashion by renewed commitment on the funding side, and also the National Defense Education Act, which opened the door to, I think, many people in this room, or the people who came before us, going to higher education and acquiring technical and scientific skills. So I think the history is incredibly important, because reminding ourselves how we got here can help us understand the current moment. And anybody who wants to stay competitive with China, or thinks that China is a national security threat, or a rival to be against whom we should compare ourselves, the answer is very simple. If you want to outcompete China, you need to out-invent them. And without invention, it's not that invention is the most important problem to be solved. It's without invention, without this scientific enterprise, you have nothing. Nothing at all. You are the French Air Force in 1923. Looks good. Congratulations. It's worth nothing.
They had lovely uniforms, though.
The hats, I think the hats of the French army were to be absolutely.
So to summarize, I hear, curiosity, willing to explore the world, how it functions, but also the sense of community, to belong into the scientific community, and an urge to also make a difference, that this knowledge is to be used in policy making, into changing the world?
At least understand the world you live in. At least be cognizant of the momentous changes that come from moving fundamental science.
But we should be careful, because we're scientists and we lack a control group. We don't have a matched group of people with similar ambitions. And I think often, honestly, even myself, of all the random moments in my career, and maybe it would have gone great no matter what happened, or maybe surely, we have this exceptional outcome, and we should believe in some degree of luck in getting us all to this stage.
Absolutely.
Yep.
Thank you. And now I'd like to open up for questions from the audience, and we have a question from the audience that are with us on YouTube.
Yes, I believe this question is for Dr. Jumper, but anybody who feels so inclined. How can AlphaFold be integrated into public health and personalized medicine?
It's a very interesting question, and I think part of, and for people in the audience, personalized medicine is really thinking about unique to a particular patient, how do we treat them? And we have a very small number of therapies right now that are very particularized to a patient and generally very expensive. I think that AlphaFold in some ways is kind of the basic technology tells us more about what's the same between many people than what's different. There are technologies that we and others have built on top of it. There's a tool called Alpha Missense that predicts for variants in someone's genome which ones might be pathogenic in a genetic disease. But I think that it still travels relatively indirectly. We still need to build technologies on top of it, that we don't get personalized medicine immediately and for free. We can start to think about things like protein design, we can think about individual variants, but it's going to have to go through more discoveries. And I think that kind of AlphaFold will accelerate it tremendously how we understand the cell. It will help us also, technologies based on or inspired by it will help us develop therapeutics, but we still have a ways to go. Let's develop, even standard therapeutics very rapidly, and then we can think about how it can be so rapid that we can do it for the patient that's just presenting. All of these, I think, are important, but it's worth also saying that we often don't understand the vagaries of diseases that we call, autism, that we call cancer, right? There are many, many things. I once heard it called the Anna Karenina principle, right? That all happy families are alike and each unhappy family is unhappy in its own way. But, in diseases, something similar. There are many, many causes of what we give common names. And I think we'll see more of that before we see even direct patient personalization, but all of this will come, and all of it will come ultimately from understanding.
Thank you. Now we have a question from Marcia McNutt, President of the National Academy of Sciences.
Yes, so I have perhaps a more philosophical question to ask you all. I'm curious whether you think that the great discoveries that have led to Nobel Prizes are the result of more or less a research system that allows for serendipity, or do you think that we would be better off if we actually had a strategy for how we fund research in America?
Very good.
So one of the joys of my life is I got involved with NASA and watched how different they do science from how NIH, which is what funds much of what I do, does science. And NASA does what are called decadal surveys, where every 10 years they go around to everybody who's studying cosmology or planets or stars or whatever, and they say what's important, and it's a whole process, and I'm sure it's politicized and people despise it, but to me, looking at it from the outside, it looks extremely rational, and they result in things like the James Webb Telescope, which is this amazing thing that they made, and it's distilled, and it's been voted on, and it's rational, but this is big ticket science. Yeah. And then my experience at NIH has been things that bubble up from beneath, and I'd like to, NIH has a huge budget. It's a visionary product of Mary Lasker sort of browbeating Congress in the 40s to start the National Institutes of Health, and it happened. But it's the surprises that come from somebody putting in a $250,000 a year proposal, and I hate this idea of administrators saying, this is what we should do. You need to do it for big things like a genome project, for sure. That was visionary, fantastic, but there's a lot of big ticket items that are not so great. And the stuff that bubbles up from beneath, that study sections judge. So I'm not answering your question because there's both I like.
The answer is yes to both, right? Yeah, maybe more rational thought and planning along the lines of strategic, what do we want to look like in 10 years? What do we want to accomplish based on what we know today? But then at the same time, there's so much we don't know. I mean, yeah, I've been funded by the NSF and also the NIH mostly, but I'm here to tell you that I almost never did what I proposed in the NIH. And so constant apologies, constant just, waltzing around,
They're going to revoke your Nobel Prize.
Yeah, but I think that's not uncommon. You know, at my institution, University of Massachusetts Chan Medical School, it's a highly collaborative place. We do projects all the time that are just totally unfunded because the institution, as it happens, has a mechanism for providing discretionary funds that people can use just like that if they want to collaborate. So I think if you took a poll, it would be fascinating to see where folks' work really was funded, but we spent the NIH money, but it was on different stuff,
And you got a Nobel Prize.
Yeah, so I think that this idea that serendipity is absolutely essential. There's more fundamental things to learn than we know, or that we could even possibly imagine, let's say. So you need to have that going, but at the same time, discussions here pointed out that you really need to think ahead, in a certain way at the same time.
I think one other point I would just make is when you think about what a strategy is, and there's no strategy that requires determinism, of course, right? Mm-hmm. But as an ex-physicist, the world itself is multi-scale. Scientific research is multi-scale, and we probably do need to think more about. We've done a really good job at supporting the very small scale and individual investigator grants. We've done a pretty good job at some really big science, like the James Webb, like the LHC. AI especially, I think, creates opportunities for mid-scale science, of scaling up ideas, things that are starting to work, into mid-sized projects, into team projects, into ones with more compute, but not enormous prohibitive going to Congress investments, but these type of mid-tier and other tiers. And I think thinking about how we fund that in the academic sphere is a really interesting policy challenge that could deliver a lot of scientific benefits, but it has to be done carefully because you have to put a lot of thought into kind of the mid-scale.
So
this is going to be the last answer now. So
okay,
yes.
I have half an hour. Is that right? So look, I just want to emphasize to everybody. This is a very deep and important and timely question. Yeah. But it's essential to frame it, I think, this way in historical context. The United States, at our peak, spent 2% of GDP on research and development in the 1960s. Now, some of that was military weapons and so on and so forth, but we currently, or of late, have spent between 0.6 and 0.7% of GDP. So as our economy has grown, the research and development spending has not kept pace. That's fundamentally most important. And if you think about what we need as a country in terms of either you could do it by sectors or potential breakthroughs or whatever, we should do everything. With 330-some million people in a world of 8 billion, we're the most innovative society. Okay, second to Sweden or third to Sweden and Switzerland. right after that, we're incredibly innovative, and we should just do more of it, and it'd be good for us and good for everyone else. And I think the answer to your question is three answers. First of all, when there's something that we can sell, you can sell to the politicians, a mission-driven research, go for it, because that's something that people can believe, they can understand, and then Victor can siphon the money off and get a Nobel Prize along the way. Secondly, I think every scientist I've ever met emphasizes, particularly the young scientists, the serendipity, the bubbling up, and the frustration of only being able to apply for an NSF grant when you've already done the research, and you're sort of understand that process. So I think the third thing, Marcia, we need to focus on, in addition to more resources at the federal level and every other level, is where else do we get the money from? So the fact that Google supported DeepMind and we have a Nobel Prize here is fantastic, really encouraging, and hopefully we could pull the private sector used to do a lot more fundamental. Bell Labs, for example, did a lot of fundamental research. They drifted away from that. So here's a very interesting example of what could change. But I think, Marcia, universities need to do more. Universities are sitting on huge endowments, which they've earned and which people gave them to support research. And what have they done for society lately? To what extent do universities generate and support? I mean, Victor gave you examples of how UMass does, but more broadly, I think we need to tap those resources and fund the science and the commercialization of it and the new job creation piece. And we need, I said I needed half an hour, and we need a technology dividend. Alaska cuts a check for every citizen every year based on oil price, oil revenues, because they have a dividend
you
model.
Want a dividend check on?
You need a technology dividend model, and you know what? Every time we win a Nobel Prize, put an extra $50 in it, or $100 for every American. right? That gets people's attention. Right, what has happened is we've drifted, people have lost that connection between this incredible research and the economic realities and the economic future. Cut them a check every year, the national technology dividend.
100% tax on the Nobel Prize. Right.
Again, one of the most influential voices in the world speaks.
Thank you so much. I take it that the message from the Nobel Prize laureates is that everyone should do more. I mean, they should donate their prizes, and the government should spend more, companies should do more, and universities with money should also do more. So more science. And with that, I'm afraid that we need to wrap up this symposium. And thank you all for attending. Thank you, Marsha McNutt and Dr. Lars Heikensten for your keynote speeches. They were really splendid contributions to this event, setting the scene about Alfred Nobel and the importance of the Nobel Prizes. And to our distinguished Nobel Prize laureates, thank you so much for sharing your knowledge so generously and also some advice for the future. Thank you so much. And please join me in giving them a hand.